Previously, we have seen how the structure of DNA and the complementary base pairings allow for DNA to be semi-conservatively replicated. Each base only binds to its complement. As Chargaff's rules tell us, adenine binds to thymine and cytosine binds to guanine. These base pairings are what hold the two halves of the helix together. This reiterates what we have seen countless times in the study of biology. Structure imparts function. Let's take a closer look at the structure of DNA and of RNA and see how this more detailed structural knowledge can help us to understand the central dogma of biology. That is, DNA makes RNA, which makes proteins. The production of RNA from DNA is called transcription, and the production of proteins from RNA is called translation. Here, we will look at how DNA serves as a template for the production of RNA, which then serves as a template for the production of proteins. The bases in DNA are more than just a template for replication. They are a code. This means that the order of the bases along either strand is critical to its function. This is referred to as the genetic code. At the most basic level, this code is read in groups of 3 bases. Imagine what a language where all sentences were made of 3-letter words would be like. This is the language of DNA. Each 3-base set is called a codon. Each codon within a gene codes for a specific amino acid. This is how the DNA code is translated into proteins. The code is read, and the amino acids are assembled in the order specified. The strand that is read is called the template strand, and its partner is called the non-template strand. It is called the template strand because, just like in replication, it serves as a template for constructing a complementary strand. In DNA replication, this is a complementary strand of DNA, but in transcription, it is a complementary strand of RNA. The production of RNA from the DNA template strand is called transcription. Transcription produces many kinds of RNA that are destined to different tasks. Here we'll be talking mainly about RNA that goes on to serve as a template for protein production. This type of RNA is called messenger RNA. However, the cell can also produce other types of RNA that can serve as things like enzymes, which would be called ribozymes, structural components like ribosomal RNA that is part of the ribosome, and transfer RNA, which plays a role in the creation of proteins, which we will look at in detail later on. Structurally, RNA is not that much different from DNA. However, there are some differences that matter. The first difference is in the namesake. That is, the ribose sugar in the backbone of RNA is not deoxygenated. The number 2 carbon in RNA is bound to a hydroxyl group and not just a hydrogen as in DNA. Also, the base thymine is replaced by uracil. We can see that the structure of uracil is very similar to thymine, and both are able to bind to adenine as a complementary base. RNA is generally single-stranded, but double-stranded RNA does exist. RNA can form hairpin shapes, where it is folded back and hydrogen bonded to itself. This is a lot like protein folding, and a mechanism involved in the formation of RNA enzymes. Just as with DNA replication, bacteria and eukaryotic cells do things a little differently. Let's look at the simplified process of bacterial transcription to get an idea of how it all works. Then, we can look at the process in eukaryotes to get a deeper understanding of how transcription is regulated. In bacterial transcription, most of the work is done by RNA polymerase. RNA polymerase is similar to DNA polymerase in that it creates a complementary strand to the DNA template. However, this strand is RNA. RNA polymerase is a holoenzyme that does the job of the helicase and the polymerase. Holoenzyme just means that it is made of different proteins bonded together in a quaternary structure. Let's look at an overview. RNA polymerase binds DNA. This is called the closed complex. Then, it pulls the strands apart to create an open complex and moves through the transcription unit, creating the RNA strand until it hits the terminator sequence, and the strand is released. The basic overview is true for both bacteria and eukaryotes. However, the details are somewhat different. In bacteria, the proteins of the RNA polymerase bind to the promoter. The promoter is a sequence of DNA that signals the start of a gene region. It is often identified by the DNA sequence TATA, or what is called the TATA box, but these sequences can vary. The completely bound RNA polymerase holoenzyme at the promoter is called the closed complex. The RNA polymerase then unwinds the DNA to create a bubble of open DNA, or the open complex. It then uses the template strand to add RNA bases in the 5' to 3' direction. Once the RNA polymerase reaches the terminator, It stops and the RNA strand is released. The terminator is a sequence that causes the RNA to hairpin because of regions of repeating cytosine and guanine base pairs, followed by a sequence of uracils, which are called intrinsic terminators. We will talk more about what happens next when we look at translation. Eukaryotes have more than one RNA polymerase. Most have three. RNA polymerase 1 and 3 produce most ribosomal RNA and small RNAs. However, messenger RNA is produced by RNA polymerase 2. In eukaryotes, proteins called transcription factors bind the TATA box. These proteins include the TATA binding protein, which binds to the DNA first, forming the pre-initiation complex, with other transcription factors and helps to separate the strands. These proteins then recruit the RNA polymerase 2, which binds and forms the initiation complex. The initiation complex then waits for a signal from an activator, which signals for transcription to begin. Activators usually sit upstream from a transcription start site and help regulate the beginning of transcription. Upstream means before, and downstream means after. Then, just like bacteria, the RNA polymerase 2 transcribes the messenger RNA until it reaches a terminator. In eukaryotes, the terminator is called the polyadenylation signal, AAU-AAA. Once the sequence of RNA is transcribed, it binds to proteins that cut the messenger RNA free. Messenger RNA molecules in bacteria are not required to stick around. In fact, they are degraded relatively quickly. However, messenger RNA in eukaryotes are often moved around the cell because of targeting signals, so they must be more durable. An unprotected messenger RNA will be attacked by defensive exonucleases that chew it up from the outside ends. To avoid this, eukaryotic messenger RNAs are sent through processing. This processing includes the addition of a 5' cap and a poly-A tail. The 5' cap is methylguanosine, and the poly-A tail consists of a long stretch of adenines that are added by the poly-A polymerase. These modifications protect the messenger RNA ends from degradation and allow them to travel to their destinations around the cell. Eukaryotic messenger RNAs experience one last step of processing before they are used to produce proteins. This last step is called splicing. In bacteria, messenger RNA are transcribed as a complete gene sequence, where in eukaryotes, they have extra sequences that need to be cut out. These extra sequences are called introns. The segments that are left in the final product are called exons. Introns are long sequences in the middle of the messenger RNA that need to be removed for the remaining message to make the correct protein. Like a movie editor cutting out the scenes that don't fit into the movie. Splicing is carried out by the spliceosome. Spliceosomes are a holoenzyme that assemble at the junction between an intron and an exon, or what is called a splice site, and folds the RNA around to connect two splice sites which are then fused together, and the intron is cut out. This leaves the properly edited messenger RNA ready to be translated into protein. The cool part about messenger RNA splicing is that sometimes messenger RNA can be spliced in different ways. This is called alternative splicing. In alternative splicing, some exons are cut out along with the introns to make a new final messenger RNA sequence. However, these alternative splice events are not random, but are tightly regulated and have been evolutionarily preserved. A liver protein involved in drug metabolism, P450, can experience alternative splicing in neuronal cells, and as a result, gain specificity for new substrates. Alternative splicing is another example of evolution using available material to increase diversity. Now that we've looked at how DNA is transcribed into RNA, let's take a look at how RNA is translated into proteins. Pause the video here, take this DNA sequence, and write down what the sequence of the messenger RNA that it produces would be. Let's take a look at all the players in protein translation. Then we can dive into the details. First, we have ribosomes. Ribosomes are protein RNA complexes that bind the messenger RNA and other components to facilitate protein elongation. They consist of a large subunit and a small subunit, both of which are made of protein and RNA. These two subunits bind to the messenger RNA during translation. Next, we have transfer RNA. As we saw earlier, the messenger RNA contains a code, written in 3 base pair words. Each of these words corresponds to a single amino acid. In order to recognize these codes, the amino acids are attached to a piece of RNA called a transfer RNA. Transfer RNAs are molecules that help amino acids attach to their proper sequence on the messenger RNA. An amino acid is attached to one side, and the other side is a sequence of RNA called an anticodon. Remember that each 3 base pair grouping is called a codon. So the transfer RNA has the anticodon, which is complementary to the codon. It can bind to it like this. Let's look at how translation works, and we will see how these transfer RNAs help translate the messenger RNA into amino acids. The basic process of translation involves 3 steps. They are initiation, elongation, and termination. Let's look at an overview. Then we can dive into specifics. The ribosome binds to the messenger RNA and helps each transfer RNA bind and connect its amino acid to the growing chain. Each messenger RNA has a start codon and a stop codon. The start codon is where the ribosome binds, and the stop codon tells the ribosome that the protein is finished. Each of the other codons stands for one amino acid. However, some amino acids have multiple codons that can code for them. Valine is coded for by GUC, GUG, GUU, and GUA, and phenylalanine is coded for by UUU and UUC. The start codon is the initiation signal, and it codes for methionine. Methionine is attached to a special transfer RNA that binds AUG at the start of translation. The small subunit of the ribosome binds near the start of the codon, then attracts the large subunit. Notice that the ribosome has 3 sites on it. Those sites are A, P, and E, and each have a special function. When a transfer RNA binds for the first time, it binds the A site. In this case, the anticodon from the special methionine transfer RNA binds the messenger RNA in the A site of the ribosome. The ribosome then shifts down the messenger RNA, putting the methionine transfer RNA into the P site. A new transfer RNA enters the A site. It has the anticodon for GUC, which is CAG, and codes for the amino acid valine. The polypeptide is then transferred from the transfer RNA in the P site to the transfer RNA in the A site to build the protein chain. Then, that transfer RNA moves to the P site and kicks the old one into the last site, referred to as the E site. The E site is where the transfer RNA is ejected from the ribosome. This process continues until it reaches a stop codon. The stop codon does not bind to a regular transfer RNA, but instead it binds to a similarly shaped protein called a release factor, which causes the now completed protein to be released. In eukaryotes, this process can occur in the cytoplasm. But wait, if we think back, we will remember that the rough endoplasmic reticulum is covered in ribosomes. That's what makes it rough. These ribosomes are there for a special purpose. They translate messenger RNA to make membrane-bound proteins, or ones that will travel in vesicles through the Golgi apparatus and be secreted from the cell. These messenger RNA have what is called a signal peptide. The signal peptide is a sequence that is about 20 amino acids long and is right at the front of the new protein. Once this section is translated, a signal recognition particle binds to it and transports it to the ER, where the protein is then translated into the lumen of the ER, or directly into the membrane. These sections of the ER then bud off and transport the new proteins to their final destinations. In bacteria, translation is coupled to transcription. So as the messenger RNA is being created, it is being translated simultaneously by multiple ribosomes. Pause the video. Use this codon chart to write down what the amino acid sequence of this DNA template strand would be. Mutations are novel changes in the DNA sequence that can occur either as a result of DNA damage or the result of errors during replication. Let's look at some of the more important mutations and their consequences. First off are point mutations. Point mutations are mutations in a single nucleotide pair. These types of mutations can result in disease, like sickle cell anemia, where a change from A to T changes the endpoint amino acid from glutamine to valine, causing the protein to malfunction. However, not all mutations cause disease, or even have any effect at all. Some are called silent mutations. Silent mutations occur in places that either don't affect the final protein product, or don't affect the function of its section of DNA. We have looked at large-scale insertions and deletions before when we looked at the recombination of chromosomes. However, this can also happen during DNA replication, causing sections of DNA to be repeated as an insertion, or sections of DNA to be lost, causing a deletion. Sometimes, insertions can cause what is called a frameshift mutation. Because DNA is read in words that are 3 codons long, if you insert one base pair somewhere in the middle of a gene, the rest of the gene is now not lined up, and often will result in an early termination or stop codon. Considering everything you have learned about transcription and alternative splicing, what do you think a gene is? Mutations in our DNA provide evolution with another source of diversity. Small changes that can accumulate over time lead to new proteins or new protein functions. Every once in a while, this is beneficial and provides increased adaptation to particular environments. These adaptations drive evolutionary change. This is why structure and function are linked. Changes in structure lead to changes in function. And these changes are the foundational bedrock of evolution and biology.